Hello, BIOL3328. My name is Tommy Liu, part of Dalhousie University's Medical Entomology lecture. Today, I'll be talking to you guys about the bubonic plague. The bubonic plague is caused by the bacteria Yersinia pestis. This bacteria is spread by fleas and is readily carried by rodents such as rats. Most often, transmission occurs in humans when a person is bitten by a flea that is infected with this infectious disease. But it can also be transmitted through the handling of dead tissue, through clothing, and is also airborne. The bubonic plague is a vector-borne illness, meaning that it requires a living host to carry it from one animal to another. More specific, the bubonic plague is transmitted through the species Xenopsilla chiopsis, which is also known as the oriental rat flea. It prefers to feed on rats and other rodents, which can carry the plague. Today, the plague is very rare. There haven't been cases reported in Canada since 1939. The bubonic plague affects lymph nodes. Yersinia pestis will use white blood cells to carry themselves to the nearest lymph node, where they will multiply. The lymph node will swell due to the presence of bacteria and endotoxins in the cell walls. These nodes become painful, egg-sized bubo. Now I will be talking about the history of the bubonic plague. The plague of Justinian in the 6th and 7th century is the first known attack on record and marks the first firmly recorded pattern of the bubonic plague. As much as 40% of the population of Constantinople died from the plague, and half of Europe's population died as a result of the plague before its disappearance in the 1700s. The Black Death, which occurred between 1348 and 1350, was one of the most devastating pandemics in human history, resulting in deaths estimated to be between 75 to 200 million people. The plague was thought to have originated in the Arab plains of Central Asia, where it traveled along the Silk Road, most likely carried by the oriental rat flea living on black rats. They were regular on merchant ships, and due to the far-reaching nature of these ships, the plague spread across Europe very quickly. The Black Death is estimated to have killed 30 to 60% of Europe's total population. Due to the very rapid infection rate at which this illness was traveling across the continent, it's been highly debated whether the plague was in fact the culprit of the Black Plague. It has been more recently thought that the Yersinia pestis bacteria had also been transmitted to the human flea, and that rats alone did not have to transfer this illness to people, but rather humans spreading it. In 2010 and 2011, DNA evidence proved that the plague was in fact the main contributing factor to mass death during the Black Death. Biological warfare in its earliest cases can be traced back to the 14th century, where it was recorded that armies were catapulting diseased corpses over the walls of towns and villages to spread the plague. In 1940, the Imperial Japanese Army Service bombed Bingbo, China, with fleas carrying bubonic plague. In the Middle Ages in Europe, massive plague epidemics killed millions of people. The plague can still be found in Africa, Asia, and South America. In the United States, the disease usually afflicts 17 people a year on average, most often occurring in southwestern United States. The causative agent of the bubonic plague is the Yersinia pestis, formerly known as Pasteurella pestis. It belongs in class Gamma proteobacteria. It's a gamma-negative rod-shaped coccobacillus, and it takes three main forms: pneumonic, sepsitic, and bubonic. The transmission of the bubonic plague is through the parasitation of rodents, primarily of genus Rattus. Its mechanism by which it spreads Yersinia pestis is when it feeds on infected rodents, then bites humans, it will transmit the virus. The flea infected with Yersinia pestis virus may have a digestive system blocked by a large mass of plague bacteria. When blocked, the flea bites a host. It often regurgitates plague-infected blood back into the wound. Fleas that aren't prone to blockage, like the human flea, may still transmit the plague by carrying bacteria on their mouth parts. The symptoms of the bubonic plague usually appear suddenly, usually after 2 to 5 days after being exposed to the bacteria. Symptoms include chills, fever, general ill feelings, headache, muscle pains, seizures, and smooth muscle lymph gland swellings called buboes, as seen in this picture. Treatment. Without treatment within the 24-hour time range, about 50% of people with the bubonic plague will die. Effective treatment requires antibiotics such as streptomycin, tetracycline, gentamicin, and doctors may also isolate the infected person if they just deem that is necessary. To prevent the spread of bubonic plague, the best way to do this is to control the rat population and by paying close attention to the disease in wild animal population. It's said that when a plague epidemic is about to hit, there will be a mass death of rats in a city or population. There is also a vaccination for high-risk workers, but its effectiveness is not exactly clear. And that's the presentation. Thanks for watching here. If you want to find out more about the bubonic plague, I have listed out some references on this slide. Thank you.